By the end of this video, you'll be able to describe the morphology and ultrastructure of the kidney and nephron, outline the process of ultrafiltration, and discuss the molecular basis of transport in each nephron segment.
Your body needs to regulate the concentration of ions, like sodium and potassium, and the volume of water it has within it, so it needs some way of getting rid of what it doesn't need without getting rid of what it does need. The kidneys take on that role, filtering out what you don't need to form urine. They sit towards the posterior, between the 12th thoracic and 3rd lumbar vertebrae, and weigh around 150 grams. Each one has a capsule holding it together, and 2 layers of the cortex and the medulla. The nephrons, the kidney's functional units, sit across both the cortex and the medulla, and dump their filtrate through the calyx to the pelvis, where it's carried by the ureters to the bladder. You've got 2 kidneys, one on either side of your body, which are supplied with blood by the renal arteries from the aorta. About 25% of the total cardiac output runs through them. The nephron can be divided into 5 main parts: the glomerulus and Bowman's capsule, the proximal tubule, the loop of Henle, the distal tubule, and the collecting duct.
Blood flows from the renal artery into the afferent arterioles, coming into the glomerulus, which is a capillary bed. Here, plasma is filtered, allowing small molecules and water into the Bowman's capsule, but larger molecules, such as proteins and blood cells, will not pass through the membrane. Next, plasma passes through the proximal tubule, where the bulk of filtrate reabsorption occurs, including water, sodium ions, glucose, amino acids, and bicarbonate. Now plasma moves into the loop of Henle, which has 3 parts: the descending limb, thin ascending limb, and thick ascending limb. Each part has a slightly different role, which we'll cover later, but generally it's involved in the concentration of urine and the reabsorption of many important ions, including sodium, calcium, chloride, magnesium, and water. The distal tubule is the final part of the nephron, which has a role in final modification of urine before it moves into the collecting duct to transport it to the ureters and eventually the bladder to be excreted. Now let's have a closer look at the cells in each part of the kidney and the ion channels they possess.
This is a cell in the proximal tubule. Like most epithelial cells, it has a polarised structure, so the apical and basolateral membranes have a different set of ion channels to facilitate the reuptake of ions from the filtrate. On the left is the apical membrane, in contact with the lumen of the nephron. On the right is the basolateral membrane, in contact with the capillaries. About 70% of the ultrafiltrate is reabsorbed in the proximal tubule, powered by a sodium concentration gradient generated by the sodium-potassium ATPase. Sodium symporters, such as SGLT1 and 2 and NaPi2a, allow the transcellular transport of glucose, phosphates, and amino acids. These symporters drive the concentration gradient for the paracellular absorption of water from the filtrate.
Further reabsorption occurs in the loop of Henle through the descending limb and up through the thin and thick ascending limbs. Their small diameter makes the study of the descending and thin ascending limbs difficult, so little is known about them. In the thick ascending limb, the sodium-potassium ATPase drives an electrochemical gradient for the reuptake of sodium, potassium, and chloride via NKCC2. Ions are absorbed into the blood via CLCK, which is regulated by Bartin. Potassium is both reabsorbed through a basolateral potassium channel and recycled through ROMK to maintain NKCC2's function. Reabsorption of sodium and chloride ions drives the paracellular reabsorption of calcium and magnesium ions.
The filtrate then moves into the early distal tubule. Sodium ions, chloride ions, magnesium ions, and water are reabsorbed into the blood here. The apical NCC cotransporter carries sodium and chloride into the cell. Sodium is carried into the blood by the basolateral sodium-potassium ATPase, providing the driving force for NCC's function. Basolateral CLCK, a splice variant of a protein found in the thick ascending limb, moves chloride ions into the blood, regulated by Bartin. Magnesium influxes through TRPM6 and TRPM7, but its point of exit remains unknown.
The principal cells of the late distal tubule are involved in sodium, potassium, and water reabsorption in the final modification of urine. Sodium-potassium ATPase generates a driving force for sodium to move in via ENaC. Potassium is secreted into the tubule via ROMK and into the blood via KOR 2.3, depending on how much sodium is reabsorbed. Using the osmotic gradient generated by sodium reabsorption, water is reabsorbed by aquaporins. The number of apical aquaporin 2 channels varies with the requirement for water reuptake. Water is then carried into the blood via aquaporin 3 and 4, which do not vary in number.
There are two types of cell in the collecting duct: alpha intercalated cells and beta intercalated cells. Alpha cells secrete hydrogen ions and reabsorb bicarbonate. Beta cells secrete bicarbonate and reabsorb hydrogen ions. The balance between them determines their eventual effect. Alpha cells have an apical proton ATPase driving hydrogen ions out of the cell and into the tubular fluid. A basolateral chloride channel, AE1, facilitates the movement of chloride ions out of the cell to move back into the cell in exchange for bicarbonate, which has been obtained from cellular metabolism. In beta cells, AE1 is trafficked to the apical membrane and a proton ATPase is trafficked to the basolateral membrane. This facilitates the secretion of bicarbonate into the urine.